First of all, when start Nanodrop system, make blank before measure the samples by lifting the arm and add 2 microliter of purified water or T buffer, depends on DNA, RNA and protein suspension, on the top of the Nanodrop sensor or pedestal. Then, close the arm and click blank at the top left of the computer program and wait 20 seconds for blank, and once the blank complete, green measure will illuminate at the top left of the computer program, and finally, lift the upper arm and dry the sensor or pedestal with Kim wipe. But, be attention please, if there are many samples, re-blank Nanodrop with purified water or T buffer every 15 minutes. After complete the blank step, select the application of interest, for example, nucleic acid button for DNA, RNA or protein quantification from the main screen, and for sample type, click DNA 50 for DNA samples, or RNA 40 for RNA samples, or single-strand DNA 33 for single-strand DNA, as showed in the next slide. Homogeneity is important for accurate measurement, due to small size of the sample, and in the sample ID box, type the name of the sample, then pipette 2 microliters sample onto the lower measurement sensor or pedestal, and change pipette tips between samples, and make sure to clean by Kim wipe the cap and sensor or pedestal between each sample reading, as exhibited in the next slide. After complete the measurement, save the data as Excel file, by selecting or creating folder to store information in any drive inside the computer, then, lift the Nanodrop, and use a Kim wipe to remove any remaining solvent from the sensor, to reduce the contamination between samples, and click Print Report, to get a table of all data. After that, shut down and close the computer to perform final cleaning of all Nanodrop surface. This slide presented the tables that is showed through work by Nanodrop spectrophotometer system.